Dear students, today we deal with the topic recessive epistasis. Last class we deal with dominant epistasis and now it is recessive epistasis. Here the recessive epistasis ratio, it is also a modified Mendelian dihybrid ratio. What is the dihybrid ratio? It is 9:3:3:1. And here in recessive epistasis, the term itself says what it is. There is an epistasis gene. So, and the effect is from a recessive gene. So, here a recessive gene which is in the homozygous condition has a role to control the expression of another gene even if it is a dominant gene. So, students, recessive epistasis means recessive gene in homozygous state has a role to control, or it has the dominating character. So, it can suppress even if that gene is in recessive condition. If that is in homozygous condition, it can suppress the expression of another dominant gene. That is recessive epistasis. So students, recessive epistasis can be explained by the example coat color in rodents. Coat color in rodents, mice, rats, etc. In rodents, the hair color, in each hair there is a special coloration can be noted. Usually the common one is agouti. A-G-O-U-T-I. Agouti color is there. What is agouti color? In agouti color, its tip is yellow, but remaining part is black in color. That is the agouti condition. Then what is the gene condition for that agouti is there must have both the gene in dominant condition, even if in homozygous condition or in heterozygous condition. So, agouti is the dominant expression of the coat color. What is the expression? That is, its tip is yellow and its other part is black in color. And another phenotype of the each hair is black. Another is black. Then what is black? Black coloration means full coloring is black. There is the absence of tip yellow color. And how the yellow pigment is absent, then the hair is black in color. Then third category is albino color, white color. Albino color, what happens? There is absence of yellow color and absence of black color. That means there is no expression of coloration, so it is purely white in color. And usually the agouti color is best for the mice to adjust with its environment. How? Because it can be escaped from its enemy or it can be acclimatized to its surroundings. So agouti is the naturally selected color and black is the second one, and the albino we can easily identify the animal. The enemy can easily identify the animal. So for the natural selection or natural adjustment, the best coloration is agouti. And here the recessive epistasis ratio, when we go through its interaction, we get 9:3:4. Last 3 and 1 join together to form 4. So 9:3:4 is the morphology. So when we cross, we get 9 agouti, 3 black and 4 albino. And the main center of this recessive epistasis is how this 4 is formed, how 4 albino is formed. Here, if it is in the dominant condition, 2 gene must be in homozygous or heterozygous dominant condition. It can be written as A dash, B dash. A dash may be either capital A or small a. B dash means dash position, either capital B or small b. Then for black coloration, there will be absence of absence of capital A gene, and the capital B either one or both the alleles may be capital B. Then in the albino case, this is the condition, that is here both if this B gene is in homozygous recessive condition, then there will be no expression of AG. That is here we study. The presence of this homozygous recessive gene has a role to control the expression of AG. So here capital A dash, that means even if it's capital A or small a, if it is small b is present, then it will be albino in color. Then the last condition, small b is always albino in color. Then we can have a look on the cross. Okay. Let us look at the ratio, how it is formed. We can select the cross by selecting a black mice with the albino mice. But students, you have to care that the selected one should be homozygous for each locus. So here black, there is small a, capital B, albino, it is capital A, small b. I have said that if small b is there is no role for capital A, so or capital A agouti, so it is albino in color. And in the cross, this is the P1 and this is the genotype. We can write this as the gamete, only one time of gamete from each parent, and we get the F1 progeny with the agouti. So that is, we have to care. The F1 progeny is agouti with the capital A, capital B, small b. And from the agouti, when we cross two such agouti, since it is, when we select two such agouti, we get from each parent 4 different types of gametes, and we can cross or we can raise the F2 as F2 racing. Here we get this checkerboard, and we can say that wherever this capital A and capital B is present, it is agouti. So we can write it as agouti, then agouti. Since capital A, capital B, here capital A, capital B, we can write it as agouti. Here agouti, capital A, capital B, here agouti, agouti. Here again agouti. You look the pattern I am writing. This way you can make it easier. Agouti. How many of agouti will be there? 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 agouti. And here it is, what is the genotype of this progeny? There will be capital A, but there is small b is there. So here we can again find out that here this progeny will be black, here black, and then here black in color. Again we can write it as these four one as albino, while this small a has a role. That means this small a, even if this small a gene is present, what happens? It has an effect to control the expression of this B. So even if there is capital B is there to express the color as black. Even if there is capital B is there to express the color as black, we can say that the progeny will not be black. Why? Because this small gene has a control to suppress the expression of capital B gene. So these progenies, even if with the smaller conditions, all are albino in condition. So students, here this is the only that we can conclude it that even if both the gene is present, then what will be the condition? If both the gene will be present in dominant state, the condition is agouti. If one of the gene is present in dominant case, if it is capital A is there, then if it is small b is there, then it will be black in color. If there is here the condition, the smallest a is there, what will be the condition? It has a control to suppress the effect of another gene. So this is the condition for writing as the ratio 9:3:4. We can write it as 9:3:4. In another way, also we can write here as, we can change the writing as, no, it is agouti. If we take this B as the expression for black color, we can write this as black. Whatever we taking, we can write, or we can modify it as such that this is, we can write it as albino, another one, albino. That means if we are taking this, considering this B gene as the base gene, or if we can consider A gene as the base gene. If any gene as the base gene and another gene as the modifier gene. So if base gene is absent or base gene is in recessive condition, here the base gene is, we can say it as small b. Then we can say that this base gene is in small b condition. Then the modifier gene is A gene. Then the modifier gene has another role. So the modifier gene, here the recessive gene or the base gene has a suppressing effect. The modifier gene has no role. So the modified dihybrid ratio as 10:3:4. So students, you have to keep in mind that of the gene, one can give the base gene and another can give us the modifier gene. So here this is the condition for recessive epistasis. So doing not memorize this checkerboard writing or something like that. You have to understand and study well. So in order to avoid memorizing the topic, I changed the position of black to albino and albino to black. That you can write to which gene as the modifier or which gene as the base gene. Okay students, thank you.